Great. Can everyone hear me and see my slides okay?
Looks great.
Awesome. Great. Thanks so much to the organizers for having me and for everyone for tuning in. As was said, I am a postdoc at Stanford. I've been there for about a year, primarily working in the lab of Andikis. Most of my postdoc work focuses on microbial metabolism, particularly in the carbon cycle and carbon fixation more specifically. Today, I'm actually going to be presenting some work from the end of my grad experience that was at UC Berkeley under the direction of Jill Banfield, looking at something quite different. Since this is a short talk, I'm just going to go ahead and do my acknowledgements. Big thank you to our experimental collaborators at the University of Tokyo. It's Masai Kono and Keiichi Inoue. And also thank you to Oded Beja, Marie Scholmerick, who's an incoming PI at ETH Zurich, and of course, Jill and the Moore Foundation for funding. So today I'm going to talk about rhodopsins. I was happy to hear them mentioned earlier in the session today, but if you're not familiar with them, they are a broad family of photoreceptive proteins that move ions across the membrane in response to light. There's a huge amount of functional diversity spread across the tree of life in the rhodopsin world. There are versions of it that transport hydrogen ions across the membrane. There are versions that transport sodium ions, chlorine ions, and really interesting, most of these protein complexes are finely attuned to particular wavelengths of light at which they operate best. So many of them are tuned to the green or blue range of the visible spectrum, others outside of that. A really, really fascinating group of proteins. And I think there are a lot of great reasons to study them. One, of course, is that they're responsible for many important cellular functions, including, as we'll talk about today, energy generation, which can be really important in aquatic environments where nutrients are quite poor. There is also a whole field of neuroscience, which we're not going to talk about today, called optogenetics, that relies heavily on rhodopsins as important tools for studying the brain. And part of that is the exploration of novel sequence diversity from the microbial world that can be engineered and developed further in lab. And today I'm going to make the case that rhodopsins are actually interesting and potentially underlying some symbiotic interactions in the microbial world. Most of my PhD was spent working on a group of bacteria called the candidate phyloradiation. I think this tree needs no introduction in this group, but this is a large phylogenetically diverse group of ultra-small bacteria, often on the order of 0.2 microns or less, living parasitic or episymbiotic lifestyles. And I have them circled here with a purple box. During the period of time that I was focusing on the CPR bacteria, we found that a particular subset of them, largely drawn from high latitude lakes in Scandinavia. This is a really nice set of samples published, I believe, as a scientific data paper by Moritz Buck in Saripura. A subset of these CPR organisms actually did contain a rhodopsin gene, which was very unexpected. And the main question really driving this study, which was published last year as a short report, was really asking a set of simple questions, right? What is the function and the ecological relevance of rhodopsins in CPR bacteria, where we don't really expect to see them? So of course we wanted to kind of move beyond our metagenomic predictions. Some phylogenetic analyses suggested that these CPR rhodopsins were likely proton pumps, but we had the really great fortune to work with some experts in this field. So this is folks from the group of Keiichi Inoue at University of Tokyo, particularly Masai and Yuma, and they performed a fairly standard experiment in the rhodopsin world, which is that you take your protein of interest, you synthesize it, and you express it in E. coli, and you basically subject these cells to light illumination, and you measure change in the pH outside of the cells during that period of illumination. So you can see 3 separate CPR rhodopsins here that were being characterized and illuminated to light. That's that line in yellow as we move forward in time along the x-axis. And you can see that under light illumination, the pH outside of the cell actually goes down. And so this is suggestive of the fact that these are indeed hydrogen ions. And if we add a molecule called, proton pumps, and if we add a molecule called TCCP, which dissipates that proton gradient, the effect is gone. So I think from this experiment, we can make the conclusion that CPR rhodopsins are most likely outward-facing proton pumps that are responsive to light. Since we're working in a metagenomics context, we actually have access to the entire genome information for these organisms, and we can look around in that genome information and find that indeed they also encode the genes for proton translocating ATPases, and that likely, like many rhodopsins that we know of in the microbial world, form a simple metabolic circuit in which the proton pump by the rhodopsin can be coupled to energy generation by the ATPase. So this is nice. It's a form of basic phototrophy that could augment the relatively simple metabolic platforms that we know these organisms to have. That's cool. But we wanted to go a little bit deeper here. And to do that, I need to give you a bit more background on rhodopsins. So as I said, right, these are protein complexes that sit in the membrane, generally composed of 7 transmembrane helices, although this is not always the case. But right in the middle of those transmembrane helices sits a really important molecule called retinol. This is kind of the essential cofactor that is light sensitive, and a change in the conformation of this cofactor is actually what's responsible sort of for the broader conformational shift of the complex as a whole, and allows it to allow an ion to pass through under the illumination of light. Many organisms that encode a rhodopsin, also nearby in the genomic context, encode the gene that is necessary to produce this molecule retinol, which is actually synthesized from beta carotene as a precursor. So again, we're working in a genomic context. We went to the genetic loci where these rhodopsins were encoded in the CPR genome. This is what I have highlighted here in orange. And we looked around for this gene called BLH, which is responsible for producing retinol from beta carotene. We actually didn't see it. It wasn't there. This is actually not without precedent. So there are another group of rhodopsins called heliorhodopsins that are found in various microbial groups. These are organisms that also lack that BLH gene required to produce the retinol cofactor. And it's been shown through some really excellent structural work that these rhodopsin protein complexes have a little hole on the side called the lateral fenestration that allows retinol that is from an entirely different source, that is exogenous, to actually enter into the protein complex after it is folded and allowed to be functional. So we thought that this might also be the case for our CPR, and Masai performed another set of experiments to try to get at this. So over here on the left-hand side, we have an evaluation of what the absorption spectrum is for our CPR rhodopsins. You can ignore that peak on the far left and note that the peak absorption for these CPR rhodopsins tends to be in about 550 nanometer range. This is kind of a blue-green that we would expect for organisms living in sort of shallow to medium depth lakes. And so the experiment that Masai performed was to actually remove retinol from these protein complexes and check that absorption spectrum again. And so that's on the right-hand plot, this curve that's in green. You can see when there's no retinol included, that absorption peak around 550 nanometers actually lost. This is not a functional protein complex. However, when retinol is reintroduced in the curves above that green one, even after about 30 seconds, but particularly after 15 or 20 minutes after the retinol is reintroduced, you can see that 550 nanometer peak is recovered. And so that suggests to us that rhodopsins from our CPR bacterium can efficiently bind retinol from exogenous sources. And Kichi went ahead and did some AlphaFold structural modeling and showed that likely our CPR rhodopsin also has that lateral fenestration that allows the binding of exogenous retinol from a structural perspective. This is really cool, and we wanted to go one step further and ask, okay, well, they're getting this retinol from some other source, but how are they getting it? Where is it coming from? The problem here is that retinol is a hydrophobic molecule, so it doesn't freely diffuse in the aquatic environment. And we figured that if the CPR aren't making their own retinol, then maybe they'd be getting it from some other member of the microbial community. And one way we thought we could test this was actually going back into our microbial community and look for that gene that I mentioned, that BLH gene that interconverts beta carotene into retinol, and ask who those organisms are, if there are any. So again, we went back to the metagenomics, particularly the metagenomic assemblies that had been generated in the process of this project, looked for any BLH gene that we could find, and then did a taxonomic profiling of the scaffolds that encoded those particular genes. This plot here that I'm showing you, each row is a different sample where the CPR rhodopsin was found. And then along the x-axis is basically the percentage of scaffolds encoding that BLH gene belonging to different microbial groups. And you can see that by and large, the vast majority of these genes required to produce retinol are encoded by the actinobacteria, which are very common members of freshwater lake communities. This was really interesting to us because we know that in other microbiomes, namely the oral microbiome, human oral microbiome, or animal oral microbiome, that actinobacteria likely are the hosts for this particular CPR bacterium that we were studying here, which is called Saccharibacterium. Here's a really classic image from a 2015 study showing this larger actinobacteria with multiple CPR symbionts on it. That's the red cells and then the green cells, respectively. And we thought that if actinobacteria were also the hosts for Saccharibacteria in this lake environment, then their interactions could potentially explain how those Saccharibacteria get this key cofactor that they need for the rhodopsin to be functional. And in fact, in the years after this 2015 study, it's been shown in some other environments that this actinobacterium, saccharobacterium relationship likely holds, at least in a general sense. So that would look something like this if we go back to our schematic. And we wanted to think about how would this transfer actually happen, right? Again, this is a hydrophobic molecule. And so we thought of two ways. One could be from direct membrane contact. As you can see on the image on the right, these two cells abutting each other is a very common feature of these interactions. And so potentially direct membrane contact followed by the exchange of some membrane materials, including the retinol, could be a way that these CPR get the retinol that they need for their rhodopsin to function. Alternatively, perhaps a vesicle could sort of bubble off of the host cell and then eventually fuse with the membrane of the CPR. We don't know exactly. Again, we're working in a metagenomic context, and so we could look at the genome of these potential hosts, these actinobacteria, and it turns out that they have their own rhodopsin as well, which is entirely unrelated to that in the CPR. So it's not a likely candidate for horizontal gene transfer. It allows us to propose an exciting model in which this host cell, which has the capacity to produce this key cofactor for photoheterotrophy, does so both for its own rhodopsin and then also that of its symbiont, potentially as sort of a form of modulation. So again, this is of course speculative at this point, and we need to do some experimental work with these organisms potentially in a co-culture setting to evaluate these predictions. But I think it's an exciting model. So with that, I will wrap it up and take any questions that people have, and go back to the screen.
Great, we have some time for questions.
Hi there, great talk. I was wondering, what is the specificity of retinol binding to different types of rhodopsins?
That's a good question. As far as I know, there isn't a huge amount of molecular heterogeneity among retinol. There's those two states that I don't think I mentioned, but there's sort of the cis and then the trans version of those things. As far as I know, most rhodopsins sort of bind a similar form of retinol, but it would be interesting if, in fact, there were sort of additional layers of kind of specificity to that. Would be something to look into. Thanks for the question.
Thanks. That was a great talk. And if I may say, all the talks today have been great. I haven't been at a conference since before COVID. This is fantastic. Sorry, enough editorializing. What are there hypotheses for what is driving the symbiosis? What sort of an exchange there might be? You might have said, if so, I missed it.
Yeah, that's a great question. I didn't touch on it. So most of the work that's been done on the CPR host interactions comes from the human oral microbiome, and which is just one of the many environments where we find CPR, and is just one, and generally focuses on one of the many CPR bacteria that we know of. So again, I think our knowledge of these interactions is still in its relative infancy and is an area where our field has to grow quite a bit. There's been some really great work that has come out again from the oral microbiome, driven by folks at the Forsyth Institute in Cambridge, Mass. And they have shown that there's sort of a number of different features that could underlie the symbiosis. I mean, just from the level of kind of attachment, right? So CPR bacteria are often enriched in pili and other attachment machinery that might actually help mediate these interactions. I believe that they also put out a paper showing some sort of like metabolic interdependencies between the host and the CPR symbiont. But at this stage, we don't really know what actually what drives these things, and there's likely to be a huge amount of variation. There's been some nice work from other ecosystems showing that CPR host interactions can be a bit more on the parasitic side. There's an organism called Vampyrococcus, which I think the name tells you all you need to know, all the way over to the other side of the spectrum, which might be more mutualistic. And I think we're likely to discover a huge amount of diversity along that spectrum of symbiosis in the future.
Are there any other questions? If not, I think it's time for a break. Yeah, I think I'm again.